uh, very kind introduction. I'll take 2 minutes to load the slides and then we can start the lecture.
Okay, sir, no problem.
Am I audible?
Yes, sir.
Can you see the slides?
Yes,
Yes.
sir.
Yes.
Can I proceed?
Yes, sir.
Okay. Thank you very much. I left active research 10, 12 years back. So I'm a fossil now. In a old age home, I'm in my Coimbatore. What I wish to convey today in this refresher course, 4 or 5 points. One, briefly we will see what is science and how it started. Was there science in this country? What are its branches? And how the history of two of the branches, chemistry and biology, led to the establishment of biotechnology. The second thing that we'll try to understand is a national level problem, which is essentially a political problem, socio-economic problem, and how scientists suggested solution to this, and that involved a lot of biology, a lot of biochemistry. We were already doing some work on this, and how we got into this, and what we did because of the directions given by government. And the third thing that I wish to tell you, that program of work which was running for years did not help for logistic problems. And so the government asked us to do recombinant DNA technology based protein hormone. I will describe the detail and the protocols and all that to solve this problem of poverty. Below poverty line, there are more than 300 million people, especially children. How this affects malnutrition and brain development and all that. And this program called Embryo Transfer Technology Program in Water Buffalo attempts to help this and solve this problem. And then I will drop a bombshell in the fifth point, how political interference and a scant regard for standard science essentially did not solve this problem. And the most important reason for this is that the politicians changed the definition of the problem. And therefore, all of us were out of business, and the poor buffalo is still suffering. And poverty has not got eliminated from this country. So I'll make all this technical, socio-economic, socio-political points and see whether I can carry you along with me. So we start with the first point. Nowadays, you will see a lot of hype saying that we knew everything under sun thousands of years back. Now, if you ask a simple question, what is science? Please remember, we refer to experimental science as science. There was no experimental science in this country or anywhere in the world till Renaissance started in Europe, mostly in Italy and then in England. And Industrial Revolution started this experimental science. There's a lot of philosophy behind it. We will discuss all this later. So this science, which we call experimental science, essentially deals with measurement. Whether you take physics, whether you take chemistry, whether you take botany, zoology, geology, or any science, what this essence of this is that you have to measure something. Without measurement, that is not experimental science. Now, this natural science And the philosophy of this natural science was given to all people by a person called Francis Bacon, who was a British lawyer and a finance minister of the then British government. And he started defining this, and he also defined what is called scientific method, how to go about doing science, what exactly science, how is it different from philosophy or anything else. Subsequently, this natural science, as it was called, earlier it was called natural philosophy, but when the experimental part was added, it is called natural science. It slowly divided itself into physics, chemistry, and biology within quotation. And very soon, each one of them developed a separate philosophy and major problems to be tackled. Physics measures operational concepts like temperature, force, voltage, etc., and hence tries to explain the physical reality of this universe by examining the relation between these variables or operational concepts. Some of you are physics people, probably. So if you open a physics textbook, you will see chapters which are called as laws of thermodynamics, laws of electricity, laws of mechanics, all sorts of things. So every chapter is a law, LAW. So physics essentially tells us the behavior of this nature under various conditions, and it seems to be an inviolable truth because nobody has violated these laws anywhere. That is why they are called as universal laws of thermodynamics, universal laws of mechanics, universal laws of electricity, so on and so forth. Meaning that even if you go to Mars, even if you go to Moon or Sun, these laws will be operable. There's no exception. That is why intellectually, among the natural scientists, physicists are the most arrogant and most strongest intellectuals. I'm not using this word in any bad sense. It is highly logical science. The second part of natural science is chemistry. Chemistry also has a philosophy. It believes that this universe is consisting of a number of materials, and each one of them is made up of some substance. However, behind all this, there is one substance, universal substance, which is transformed into many materials and all, fooling us. And therefore, philosophy of chemistry for centuries searched for this universal substance. Later, we realized when periodic table came that there are fundamentally 70 and odd substances. We call them elements. And later, this list is expanded. We have 100 and odd. That's not the point. The point is that these fundamental particles or fundamental substances are the ones which constitute this universe. And the relation of the structure of each one of them to the function is the main duty of a chemist to understand. The third one was biology. I'm using this word. Biology is a strange word. Many people don't understand what is this word. Biology first committed a crime because biology did not measure anything for more than 1000 years. I'm not exaggerating. Most of you read history and you will always be told that Aristotle did everything on earth. And that Aristotle is called father of everything. Father of genetics, father of parasitology, father of paleontology, father of this, father of that. I don't know who is the mother, but at one time he had 6,000 research scholars. I'm sure he wouldn't have given fellowship to them. Imagine present day PhD students struggling for fellowship, and this guy maintained 6,000. And he asked them to go around and describe anything they saw and wrote all that and published everything under his name. That's why people in the textbooks write, Aristotle is a great man. Not at all. And he didn't measure anything. We already made a statement. Science without measurement has no meaning. Later, a French mathematician, René Descartes, he advised all people, scientists, that unless you measure something, unless you do experiment, you will not progress, you will not understand. And this was applicable to biology especially, because till then, biology was treated as a telephone directory, information. There was no question, there was no answer. Therefore, because the information load was too high, pretty soon biology teaching essentially divided into two departments, zoology and botany, and subsequently microbiology, based on phylogenetic groups, because the subject matter was only description. It is only under the advice of René Descartes, when biology started doing experiments, experimental biology came, then it became, they realized that they never developed a biological method. Even today, whatever experiments we do in biology, the methods have come from physics, chemistry, and now mathematics and computer science. There is no biological method, including surgery. Therefore, with that poor state of thing, they had no choice but to use physics, chemistry, mathematics, computer science to investigate biological problems. And that became what is called second type of biology, or reductionistic biology. The first type is descriptive phenomenological biology. It has got another name called natural history. Some of you have studied all these things. Great areas like taxonomy and ecology and other things came on this area. And the second one is reductionist biology, where you reduce biology to physics, chemistry. And therefore, subjects like biochemistry, biophysics, all these, physiology, all these things developed. It is now fashionably called molecular biology. Whatever it is, the point is that this reductionist biology did not make us understand biology, nor the class 1 biology or the so-called in vivo biology, organismal biology, that also did not make us understand. Because when you're dealing with only information, but no question, no experimental approach, you will not understand the subject. Particularly biology, which deals with living organisms, there is one property of living organisms at every level of structural organization, which people hardly mention in textbooks. I'm sure all of you have read more than me. You don't see the word emergent property in any biology textbook, zoology or botany or microbiology or anything. It is a tragedy. The most characteristic feature of living organism is emergent property. What is emergent property? A property of a whole which is not found in the parts. Which means, if I put it very crudely, in arithmetic, when you say 2 plus 2, it is 4. And nobody will question you. But in biology, 2 plus 2 is 5. What is this 5? The fifth one is the emergent property. Evolution is an emergent property of underlying cellular processes. Cell secretion is an emergent property of underlying cellular biochemistry. And therefore, what you see at the whole is not seen at the parts. That is why we say in biology, whole is more than the sum of the parts. And nobody will teach you all these things because people have forgotten the essence of biology, as they are already divided as zoology, botany, microbiology. Anyway, today we deal with all this, and this reductionist biology became the biology in due course of time. But as I said, we still didn't understand. We will complete this story in some other lecture, but now let us take this, the other point that I was mentioning. Natural science has physics, chemistry, and biology, and chemistry is one other branch of natural science. And what does this chemistry do? I have already mentioned the philosophy of chemistry. Now, if you trace the history of chemistry in one slide, you notice that chemistry started as inorganic chemistry. Those of you who are old enough would have gone to old universities, you would have seen Department of Inorganic Chemistry. This was because these chemistry people dealt only with inorganic substances like sodium chloride or copper sulfate, acids and alkalis, and they did not talk about living organisms because they were afraid, and as scientists were those days, that all this is creation of God, and therefore we should not touch any living organism, do any experiment. We don't even understand them, so leave them. So let us deal with inorganic. That's how inorganic chemistry came. And then slowly people said, these living organisms, do they have the same elements and compounds as you see in a piece of earth or soil? A very curious question. And so some people dissected living organisms and did the elemental analysis, and they found to their surprise that the same elements that you see in air or water or soil sample, all these elements are present in living body also, whether it is a microbe or whether it's animal or whether it's a plant, no difference. Then why are these living and what are the others are not living? Then they raised the first question in biology, what is living? Till today, we don't have a good answer. I will tell you all this later. Now therefore, the chemistry people realized that we can get elements and compounds from nature also. At that point, the word nature did not mean sunset and flower or anything. It only meant plants, trees. And therefore, they got these chemicals from plants, and therefore chemistry, inorganic chemistry became natural products chemistry. If you go to Delhi University, if you go to Calcutta University, if you go to Madras University, you will see very well developed natural product chemistry labs. And they exhausted everything. And then they asked another curious question. That's how when you ask a question only, you will get the answer. Without question, you don't get answer. So they asked suddenly an embarrassing question. What are they doing inside this thing? Can we synthesize? Now, when you say I can synthesize, you are dropping a bombshell. Chemistry people knew how to synthesize inorganic things. And when they found these natural products from plants, millions of them and variety of structures, they said, can we synthesize? And they said, no, we cannot synthesize because this is all God made. At that point, Frederick Wohler synthesized urea, which is present in our body, in our urine, in everywhere, any living organism. They synthesized it by taking an inorganic substance like ammonium cyanate, silver cyanate, as the books write, that is wrong. He started with ammonium cyanate, converted into, heated it, and then got what we call urea. Urea is the fertilizer. It is found in our body. Every day, all people excrete at least 30 grams of urea in their urine. You read any clinical biochemistry, you will get this. And therefore, they said, this was a philosophical breakthrough. And therefore, they said, we can synthesize even compounds which are found within living organisms. You don't need God for all this. And therefore, natural product chemistry became organic chemistry. Now they asked the third important question. What are they doing inside the plant, inside the flower, inside the liver, inside the brain? What are they doing? And that came another branch called biological chemistry or biochemistry. So all these things, essentially then penetration and growth of chemistry into biology, and along with biophysics, this is called reductionist biology. You deal with biological things, biological processes in chemical terms, in physical terms, in mathematical terms, and that becomes reductionist biology. There is one drawback in reductionist biology. All the biochemistry people, if they know their biochemistry, they realize that they cannot explain emergent property. Nothing. We'll use a simple example. Let us use the word mind. All of you know what mind is. And if somebody asks you, show me your mind, you will show your head. But if you take a sheep or a cow or anybody and then break the skull and open, you see brain. You don't see mind. Mind is an emergent property of brain. It is not the brain. And therefore, this you will not understand if you follow only biochemistry and molecular biology. And therefore, reductionist biology did not understand biology. All right. However, because of the advice given by so-called René Descartes, biologists also said we should be useful to society. And so animal husbandry and agriculture started. These are called the applied aspects. And all the universities also started departments like applied physics, applied chemistry, applied mathematics, like that. So here is an example where I show you this Frederick Waller's synthesis of urea starting from ammonium cyanide, but he started with silver cyanide, but then converted into ammonium cyanide and then heated it and got urea. This is a classic paper, classic work. He was a student of Berzelius, and we told his teacher that I have synthesized urea. Berzelius said, "What kind of a chemist you are? I don't believe your work." That is the kind of ridicule they faced when they were doing remarkable research. Same thing continued, and you know now today that organic chemistry is the strongest branch of natural science, and they can synthesize anything. Here is few examples just to drive home the point. Erythromycin or any antibiotic or any recreational drug, anything can be synthesized. So if you summarize, chemistry gave new materials. In many universities, it is called Department of Material Science. You coin new words, but tell the same old wine. The nano revolution is here to stay. Biochemical reactions can be understood through chemistry. Biochemical transformation have helped biotechnology. Green chemistry has helped enormously. Chemistry helps in dissecting biological problems. And therefore, these two branches of natural science, biology and chemistry, merged under the advice of René Descartes, and a new branch called biotechnology came. And this biotechnology, therefore, is a direct offshoot of reductionist biology. Of course, it needed engineering for scaling up. And therefore, if you go to England, most of the biotechnology departments are found in polytechnics. You need a strong engineering, chemical engineering and plant design and all those things, plus management, plus intellectual property rights and all that. But in our country, as usual, we start everything in a haste. So we started biotechnology in the university system. There was 57 universities. Today they have MSc biotechnology. There is neither biology nor technology in those departments. That's why you don't see a single patent, single product coming from this country. This is immature way of doing. This is another lecture I will give you what we should teach. That's a different story. Endocrinology, another branch of zoology or medicine, along with recombinant DNA techniques, gave rise to reproductive biotechnologies, and that's where we made some contribution. Now I'll come to the second point, which I said I will deal with to introduce the technical work. India has a number of problems. Under previous Congress government at the center, Rajiv Gandhi days, there was an advisor called Sam Pitroda, and he identified 12 central problems for the country. These are called technology missions. One of them dealt with nutrition security or food security in another way for our poor children. At those days, about 1/3 of population was below poverty line, BPL. And therefore, and as people know with experience, that if you don't have good nutrition in the young age, you don't become a healthy person in the adult. Not only that, from nutritional biochemistry, we know that brain development is maximum during the first 5 years of postnatal life. And therefore, those poor children in villages, especially in Orissa, in Koraput and other villages, Ganjam, children are born with malnutrition and therefore they don't become good scientists or good doctor or anything. And the society simply knows how to laugh at them. It is not their fault. And to solve this nutrition problem, the best nutrition for this is to give milk. Now, here is a tragedy. India is number 1 in the total milk production in the whole world. 150 million tons of milk per annum. Then why we have poverty here? Because all this rurally produced milk comes to cities and you and I take it by pressing a button. Mother Dairy or Amal or whatever you call it. The rural people who produce this, actually they don't give it to their own children. That's how malnutrition is found in this. And therefore, people said, let us tackle this. And there was for some time, a political situation came and a political solution was also given by one of the earlier prime ministers we had, Mrs. Indira Gandhi. And she started a slogan called Garibi Hatao. It's a tragic slogan. What she did was, according to WHO definition, if you can't have money to buy 600 calories of food per day, you are called a poor person. This is a definition, WHO definition. And therefore, on that basis, about 300 million people of this country were, 1/3 approximately, were below poverty line. And this great prime minister coined this word. And what she did was, she changed the definition and made it 450 calories of energy. And therefore, overnight, 150 million poor people became rich people. That is political tragedy. There was no experiment done, no science done, no research done, but poverty eliminated. This is political slogan. So be careful about all these things. Anyway, one of the technical solutions to solve this was, even though we have the highest amount of milk produced per annum in the whole world, the distribution, there is a mistake, there is a problem. And per capita production is also, there is a problem. That means if you take the best buffalo in the country, Haryana, we have a Murrah breed of buffalo, it gives you 3,000 liters of milk per lactation. Okay. Now you go to America, USA, especially Texas, and look at their cows, they call it cow. What we have here is not a cow, it is a zebu. Now that cow, you ask that American farmer, what is that animal? He will give you a very strange answer. He can't even pronounce. He calls it Brahman cow. It was imported from India. There is a breed called Ongole breed. Some of you may be from Andhra. So you know there is a place called Ongole. It's a beautiful breed of cow, which is almost not there in India. And that Ongole cow is the best cow in the whole world. They have taken it, and by continuous breeding, we'll discuss that later, they made it a high yielding, and that gives 9,000 liters of milk per annum, per lactation. Our best breed of buffalo, 3,000. You see, our own cow, they have made it more efficient. And this has come because of a technology called multiple ovulation and embryo transfer technology, in which we made some contribution, and that is what we are going to discuss. And therefore, I'll have to tell you the third point, the hormones that are required for this, we produce, and I'll give you a 5 minutes introduction to these hormones, and then we'll go to the actual work. I don't know how many of you are botanists, how many of you are zoologists and all that. This is the tragedy. Probably there is no biologists in your audience. This is a cross-section which shows in a mammalian animal like a rat or elephant or human being. If you take section and you see here a picture which tells you what is hypothalamus, which is in the brain region on the top. And below that, there is something called pituitary, technically called hypophysis. And there is a stalk which joins these, which you see here, both nerves and blood vessels you will see. And that is called hypothalamo-hypophyseal connection. Now, why I'm showing this is, through that, some hormones from hypothalamus, peptide hormones, that's why I'm using chemistry background a little bit, and which stimulates the pituitary, especially the anterior pituitary, technically called as adenohypophysis, and that in turn produces a number of protein hormones, about a half a dozen. We don't have to go into detail. 3 of them are very important: follicle stimulating hormone, luteinizing hormone, prolactin, and thyroid stimulating hormone. 4 of them, they are very important for reproduction function. All physiological processes which are clubbed together as reproduction. So which includes in mammals, milk gland and milk production, gametogenesis, spermatogenesis, oogenesis, and all that, and the further process of fertilization, embryo development, implantation, all these things which you read in reproductive biology, all these are regulated by these hormones. So we have to deal with these hormones, and our lab was a pioneer in choosing the animal model, which is buffalo. At that point, I was a lecturer and everybody was laughing at me. Why you are not working on Harvard mouse? I chose buffalo as my animal. And we did a lot of work, got a lot of recognition and all that. However, we were still humble. Somewhere in late 80s and early 90s, this embryo transfer technology program was identified as one of the technology missions. And then buffalo became the identified target animal. And therefore, buffalo became very famous throughout India. And therefore, Muralidhar also became very famous because till then we were only doing science. And then we started politics. I'll tell you the detail.
Last class free books percentage of films are.
You were asking something? Hello?
No, sir, their mic might be on, sir. No problem. You continue, sir.
Okay. As I mentioned just now, this hypothalamus, hypothalamic peptide hormones, neurohormones they're called, they come and act on variety of tissues in our body, especially pituitary, thyroid, adrenal, bone, gonads like testis in the male, ovary in the female, kidney, uterus, everything, and in turn regulate the physiology. Therefore, these hormones from anterior pituitary are very, very important, and we did a lot of chemistry of these protein hormones. I will not go into detail, but that was very helpful. And what was the work done? Biochemical purification of all these hormones, especially buffalo follicle stimulating hormone. As the name says, it stimulates ovarian follicle growth. And we did this. Okay, if I summarize this work, how much yield you will get? That's what we reported, 3 milligrams per kilogram of pituitary. Now I will give you some data which will tell you what this means. The pituitary gland of buffalo, we used to go to Aurangabad, where they have the abattoir where they slaughter 3,000 buffaloes per day. Me and my student used to go there with the eggs and a lab attendant. When the head comes, we used to break the skull, then remove the brain and look at the tellus turcica and remove the pituitary. Immediately put it in liquid nitrogen, bring it to Hyderabad, purify hormones and all that. That's a technical work. And the yield we get, when we say pituitary of buffalo, it is about 1.2 grams. All right. And 1 kilogram of pituitary means 800 buffaloes killed. How much you get? FSH, 3 milligrams. This is the highest reported in the world. So we are not bad. But is this 3 milligrams enough? We will see it is not enough. How do you purify? We will not go into detail. We have to use what are called purification protocols. And these are basically biochemical methods or affinity methods or scaled up methods. Here is one diagram to tell you one sample how to purify a given hormone, say LH or prolactin or FSH or whatever it is, by extracting or starting with pituitary and going through all these steps. You don't have to read all these things. I just give you the complication, how purification means a lot of biochemistry work, lot of chromatography, lot of separation techniques, lot of things there. Let us say we did all that and you get these hormones. And then the chemistry people will say, how do you know you got a homogeneous preparation? And the biochemistry people use the word called purity when discussing homogeneous preparation. What is purity? Purity is not religious purity here. We are not talking about it. It means the substance that you got is homogeneous and only that substance is present in your hand or test tube. And for that, there are techniques. Some of you know all this. Maldi-Top spectrum of monomeric prolactin. This is how you get it. And a single monodisperse peak means your substance is highly pure. We did this for FSH, we did this for LH, TSH, everything. I'm only giving you one example. And therefore, we are competent to purify these hormones from these slaughtered animals. And you also have to show that it is not just a protein or a chemical, it should be biologically active. So there are what are called bioassays to determine their potency. We did all that under grants from ICAR and all that. This comes under animal husbandry, and we showed it is biologically active. One of the assays is called Steelman-Pulley assay, as it is shown here. You have a dose response curve with a standard hormone like human menopausal gonadotropin, and then give your hormone that you purified and estimate the biological activity. That's what we do. Every laboratory does. We did all that. This is about 20 years of work and published a lot of things. But was it interesting? Was it useful? It is not useful to anybody except for my promotion. And therefore, we were almost in a depressed state. At that point, the government of India identified MOET program for increasing the milk yield of buffalo. And therefore, we look at this program, what is to be done. What you do is a very simple veterinary technique. You take a she buffalo that is female, let us say it is January, and you take that female and give an injection of follicle stimulating hormone, if you have. And at that time, ours was the only laboratory in the whole world which had produced FSH from buffaloes. And therefore, you believe that you should inject buffalo FSH. But the people who got the grants, agriculture universities, veterinary universities, and other people did not have 1 microgram of buffalo FSH. They imported pig FSH and injected into buffaloes. Some of you know a little bit of immunology. I'm sure about it. And therefore, you realize what you get is not ovulation, but antibodies. That was a national scandal. 100 crores has gone into this program in all our universities. Not a single ovulation, super ovulation was reported. Total failure. Because they failed to inject buffalo FSH because they didn't realize basics of immunology. But they were cheating the government. Now this program, therefore, has to induce super ovulation in a given buffalo. And they gave stories to the government saying that in January, we will give an injection of FSH to this animal, she buffalo. And then after some days, it will induce super ovulation, which means number of eggs will come. Buffalo is a monotocous animal, which means it releases 1 egg per estrus cycle. You will get 6 to 9 eggs as they reported, they expected, and then collect them and then fertilize them in a petri dish. This is called in vitro fertilization. Where do you get the sperms? We have buffalo bank, sperm bank, bovine, everywhere we have in the agriculture universities, ICR divisions, we have sperm bank. And so you take sperms, fertilize them, and once they are fertilized and divide and become a blastocyst at the 16 cell stage or 32 cell stage, you put them in a surrogate mother. What is a surrogate mother? A very old buffalo, which is past reproduction, but you put these blastocysts into the uterus, give an injection of progesterone, a steroid hormone, so that the uterus will not contract, and therefore this surrogate mother will hold the baby. And then at due point, it will deliver. So what you got? From 1 she buffalo mother, using a surrogate mother, by inducing superovulation, you are producing 6 buffaloes from 1 buffalo in 1 month. After 9 months, but let us say per month. Now in February, you repeat the experiment. In March, you repeat the experiment. So if you do a simple calculation on the paper, you will see that 96 buffaloes can be produced from one buffalo in 1 year. All the great scientists, they sold this story to government, 100 crores poured in, that these people will change the scene of India, eliminate poverty, because we will increase per capita milk production by reproducing elite buffaloes, Murrah, and so everything problem is solved. Nothing happened. And therefore, now I will tell you, organization which was planning all these things, what are the advantages of all this embryo transfer technology? There are some other advantages, not just milk production. If you can do twinning, you can do sesquiplates, you can do quadruplets, you can do anything with this. Supposing you are doing this on a race horse, imagine, betting is done on race horses, millions and crores of rupees. You can produce twins from these, and you believe horoscope, and therefore you say the uncle has won that race, and therefore this child also will do. Therefore, they employ this technology in race horses. All these are science fiction. It is only written in books. Nobody has done all this. Okay. Now at this point, Government of India, Department of Biotechnology, they keep a number of foreign advisors who come to Delhi and advise them what is to be done for any technical problem. So the Secretary of DBT, no names, told them that we have spent more than 100 crores on this embryo transfer technology program, but no result. We are not able to do anything. Do you have an advice? One of the Canadian experts who came to Delhi as an advisor, he asked the Secretary DBT, have you talked to Professor Muralidhar? And all hell broke, because the DBT secretary asked, who is this Muralidhar? I was next to university, there only, Delhi University, neighbor, professor. They don't want to talk. That is the problem with this country. Anyway, then the secretary came to me and said, can you produce these hormones for us? You have to do service to the country. You must be patriotic. So I said, please don't use such strong words. I only know how to do science. And you want to produce these things? There is a problem here. I can produce and give you. In fact, they gave me a grant of 35 lakhs and I produced these hormones. And if you go by Sigma catalog prices, they were worth $40 million. Probably I am the only scientist in this country who took 35 lakhs of rupees and gave back DBT $40 million worth of chemicals, including hormones and other things. Okay. But the summary of all this is, I told the secretary, the yield of this FSH, which is required for superovulation, is 3 milligrams per kilogram. I have shown you the slide. And therefore, this biochemistry will not help us. Then the government asked me, so what should we do? So I said, you should do biotechnology, recombinant DNA technology, clone the genes for all this, express them, and then scale it up and then do it. So they immediately asked all the biotechnology departments of this country. All of them refused. They were all dealing with biotechnology department. None of them had done any work. So the government came back to me. I was a poor zoology professor. My degrees are in chemistry, but I was chairman of zoology. That is the greatness of Delhi University. So they came to me and said, can you produce this by recombinant DNA technology? I said, I am not a molecular biologist. I am not anybody. I know a little chemistry. That's all. They said, no. You learn and you do everything. Okay, challenge. So in 3 years, we learned all these techniques. We produced recombinant FSH, prolactin, luteinizing hormone, TSH, every damn thing we have finished, published it, scaled it up also. And then I told the government, now you can give it, but I don't keep buffaloes in my department. Mine is a zoology department. You ask all the veterinary departments, there is a National Institute of Buffalo Research in Hisar, Haryana. And ask them to inject buffalo FSH. I will give the FSH hormone free because I have already made a lot of it. And then after 1 month, that government came back to me and said they're asking 3 crores per injection. You see the scientists now in their true colors. Veterinary scientists who are to inject buffaloes, 3 control and 3 experimental, were asking 3 crores project. The hormone is given by Muralidhar free. The syringe costs 2 rupees. Buffaloes they have. What is the cost of the project? So the government folded its hands and said, Professor Muralidhar, you do this work. I said, I won't do this work in my department. It's a regular department of zoology. I can't do all this, but that is the tragedy. By the time, of course, I was close to retirement. And one of these days, when my last student was presenting something, and then one examiner who came, one of the famous veterinary professors from one of the agricultural universities, he said, why should we do all this MOET and all that? I said, that is what you started, the superovulation and all that. Why you should not do? Because we have not achieved high milk yield even today. Then he said, no, we have solved the problem. What is that? He redefined the word superovulation, a full professor. He later became vice chancellor. And that he defined superovulation that if you get 1.3 eggs, that is superovulation. Any textbook will tell you superovulation means beyond the number normally given, which means if this is a monococcus animal, 6 to 8 eggs, if it gives, that is called superovulation. And 20 years we were all chasing that, and therefore doing all this work and publishing and producing. And here comes the scientists who changed the definition, brainwashed the government, and government closed the whole program. That is the end of the story. I'll just give you some detail so that you see in our own career, we have seen enough politics, enough good science, enough wrong science, enough every damn thing that happens only in this country. Because we don't have commitment, we don't have conviction, we do research not for conviction, but for convenience. And that is the tragedy. This is the biggest example that I'm telling you from our own work. Now, what are the difficulties associated with this experiment, MOET, if you want to apply it to buffalo? What difficulties we have? Unresponsiveness to exogenously administered hormones. Normally, this buffalo, all of you know it, it's a very lazy animal. It is also physiologically lazy. When you give injections, it does not respond very well, like a dog or a rabbit or anything. Use of non-homologous gonadotropins. I already mentioned how the Indian scientists, politicians were injecting pig hormone into buffalo. They were getting antibodies and they didn't get any superovulation. And that is called technically non-homologous hormone. Buffalo, if you want to handle, you have to inject buffalo hormone, not pig hormone. Another major obstacle is superovulatory response variation in buffalo. Like that, there was a lot of things, and therefore, the solution was that you have to use biotechnology, recombinant DNA technology. And I already told you that there is no single report of using buffalo FSH in buffaloes, either native or rDNA. And therefore, we started this rDNA work and produced, characterized recombinant buffalo pituitary gonadotropins, FSH, LH, TSH, and prolactin. I'll take 5 minutes to tell you the techniques and what we got and all that. So you have to make a good RNA preparation, then using reverse transcriptase, make the first strand of cDNA for the particular mRNA using proper primers, characterize those that you got cDNA pieces by electrophoretic techniques, show that the size matches with known sequence and all that. And then we produce this. Incidentally, this FSH, LH, and TSH are called heterodimers. There are 2 proteins there, 2 polypeptides. alpha and beta. Together, they become a hormone. Individually, they are not hormone. All right. Now, this represents the alpha subunit, which was not only cDNA produced, but also induced. And by Western blot, we showed that this is alpha subunit. Incidentally, alpha subunit is common to all the three hormones, TSH, FSH, and LH. Read a little bit endocrinology, you will understand all these things. And then we produce beta, which is the nucleotide sequence. From that, by normal, everybody knows all these techniques, alpha and beta, all these are deposited. And these hormones are heterodimers, as I showed you, as a protein. They have alpha and beta subunit, and both of them should be together to get a hormone. But I already mentioned that alpha subunit is common. Yes, it is the beta which is different, hormone specific. So beta of LH, if you combine with alpha of FSH, you will get LH only. That is the endocrinology fundamental paradigm. So we did all that. Initially, to learn the technique, as we did not have any training in recombinant DNA technology, we only knew a little zoology, a little biochemistry. We produced this in bacterial expression system. And as you know, you can express and get the protein. It is active in vitro by immunoassays and other things. But in vivo, that means if you inject into a mouse or a rat, it is not at all active. Therefore, bacterial expression, we were learning actually, is not good for producing these hormones, which are glycoprotein hormones. Bacteria don't have glycosylating enzymes in their biochemistry. Therefore, we have to use mammalian systems or Pichia system. Pichia is a cousin of yeast. Saccharomyces cerevisiae, there is another cousin called Pichia, and you can use that for back expression. And that's what we did. But to show that we can do all this thing, we did for another hormone called buffalo prolactin before we attempted on FSH. And here is a bioassay for that recombinant buffalo prolactin. And parallel, you will also see standard native prolactin. Both are parallel. Therefore, and in fact, the recombinant one is more active, which means we also learned in this game how to unfold a protein and fold a protein to biologically active form. This is a very great game in biotechnology companies. They hide and tell you that these are all secret. It is nothing. We learned and we mastered all these things. We can fold any unfolded protein. We did a lot of biophysics on this and learned the technique ourselves. All this knowledge which we learned during career, we applied it and then applied it to produce recombinant buffalo FSH, TSH, and LH in both mammalian cells, CHO, Chinese hamster ovarian cells, or another type, Pichia, which is a yeast type cell. Both are mammalian. You need a vector. We used different expression vectors, and we put all these as cDNA. We did number of models where both the cDNAs are in tandem, and in another model, they are not joined, but in separately. In both the cases, we got biologically active hormone. This is one radioimmunoassay for TSH recombinant. There are a number of bioassays, and we showed it can be done, and we showed it also. However, the problem was, as I said, the amount you need is enormous, and therefore people said you should do a long-acting. What does that long-acting means? The carbohydrate of these hormones should have a lot of sialic acid. If it is a lot of sialic acid, N-acetyl neuromonic acid, it will have plasma half-life more, and therefore it will be biologically more potent. Animals like buffaloes don't have enzymes to put sialic acid. So we expressed it in CHO cells and produced it. Not only that, we did a little engineering at the cDNA level. We took the C-terminal 35 amino acids of HCG, human chorionic gonadotropin from pregnant woman, and as a cDNA, we joined it with the cDNA buffalo FSH beta with a glycine junction corresponding DNA, and then expressed it and got long-acting FSH, buffalo FSH. And that was my last student from Bangladesh who did PhD on these things. And this is the picture which tells you, you take the C-terminal end HCG is also a gonadotropin, but from pregnant women. It comes in the urine. And that the last 35 amino acids is called C-terminal peptide. We clubbed it with buffalo FSH, which is a pituitary hormone, and produced the hybrid hormone. This is details of the technique that you don't have to worry. We produced that and showed you can get expression and also biological activity. Everything we showed, estradiol production, cyclic AMP production, every damn thing we have done. For want of time, I'm not saying. The summary is that we have produced recombinant long-acting buffalo FSH, which, if it's injected, you should induce biological activity, which means superovulation, and therefore take up this embryo transfer technology program. Two things happened. One, I retired. And second, the government changed the definition of superovulation. So the whole program is a waste. It's a national disaster, except for the work that we did from our laboratory in Delhi University and Hyderabad. Another thing that we did, I'll take only 5 minutes. There is another hormone, just like HCG, the pregnant horse, or it is called mare, it produces a placental hormone called equine chorionic gonadotropin, which comes out in the blood. It is called PMSG, pregnant mare serum gonadotropin. Unlike HCG, it does not come in the urine. Even today, it is a mystery why horse kidney cannot pass this one. Now, this PMSG is notorious. It has got both the activities of FSH and LH. In horse, it is LH. But if you give it a rat or an elephant, it is FSH. And we showed that it is not so, that in heterologous animals, it is both FSH and LH. That's what we did. And so the problem was, can we get a LH-free PMSG or FSH as PMSG? We did that. And chromatographic separation. The second peak that you see here is a PMSG without LH activity. Why I am saying this? If you give PMSG to buffalo, you will get superovulation. You get a hyperemic activity, entrapped ovum, and therefore that hyperemic response is due to LH. So people were asking for 100 years, is LH and FSH activity in a PMSG on the same molecule or they're different? There was no answer. We separated here by chromatography. You see here PMSG on chromatography, two peaks. The first peak of PMSG had both the activities. The second peak of PMSG, smaller one, had no LH activity. Therefore, this is a pure FSH-like PMSG. And this is most useful for superovulation. We also did some molecular docking and bioinformatic work, where we showed that FSH interacts with its receptor, and mutational analysis we did and got a superactive FSH, which has got a higher affinity for its cognate receptor. Therefore, these are all bioinformatic work. You don't worry about detail. So all these essentially tells that we did three things: a long-acting recombinant buffalo FSH, and earlier to that, biochemically purified native buffalo FSH. We also did the LH-free pregnant mare serum gonadotropin, also useful for superovulation. And we also did a in silico generated triple mutant with a high KD for receptor. These three should be useful in inducing a successful MOET program and solve the national nutrition security problem and the so-called poverty problem. So I told you all the politics behind it. Nobody is working on these things now. I am retired and tired, really. I am 75. Government is not interested because somebody changed the definition of superovulation. So what we have is a breed of dishonest scientists who took 100 crores and didn't deliver anything. That's why when you do research, promise less and deliver more. Don't promise a lot and don't deliver much. That is not correct. So that's why I said truth cannot be made a casualty in any solution to a conflict. Thank you very much. I'll be happy to answer questions.
Okay, now the topic is open for discussion. If there are any clarifications, you can discuss with sir, not only with the topic, even general aspects, particularly related to syllabus and all those things. He is very much keen and because he is one of the panel member of the developing the